let me welcome everyone to the Society for Marine Mammal Editors Select Seminar Series. This is, my name is Daryl Boness, and I'm the Editor-in-Chief of Marine Mammal Science. So I don't know, this is probably about the 6th or 7th presentation we've had in this series. The series, speakers for this series are chosen by the Board of Editors for the journal and are selected from either recently published papers in an issue of the journal or papers that are on early view. Before I introduce our speaker tonight, let me just deal with a couple of practical matters. One, the presentation tonight is being recorded, so you should be aware of that. That recording will be available through the Society's website in a couple of days' time. There are a limit to the number of people who can join on Zoom of 100, although we're under that limit now, though I understand we had over 100 registered. But you can also come into the presentation through Facebook Live. I guess that's all I had in announcements. Oh, just one more. I have posted in the chat the link to the paper that's the basis behind the presentation tonight. And that paper will be available as open access through the month of August. So if you aren't a member of the Society and want to obtain a copy of the paper, you can do so until the end of the month without cost. So with that, let me go ahead and begin introducing tonight's talk. Tonight's presentation will focus on rather charismatic marine mammal species that's found globally, and that's the killer whale. Many populations of this species are in considerable danger, mostly due to small numbers remaining often from human-related activities. Effective management requires good data on population structure. While there are good data on population structure in parts of the world, there are other parts where the information is limited, and that includes the waters around New Zealand and Australia. So tonight we're going to hear about results from recent DNA analyses done by our speaker and her colleagues. And the information will add to the base of knowledge that's available for proper management and conservation of this species in that part of the world. So our speaker tonight is Ms. Isabella Reeves. Ms. Reeves is currently a PhD candidate at the Cetacean Ecology, Behavior and Evolution Lab and the Molecular Ecology Lab at Flinders University in South Australia. The paper on which her presentation is based is, interestingly, Isabella's first publication where she's the primary author, and impressively, it's based on her undergraduate honors work. The paper is not yet assigned to an issue of the journal, but it is available in early view, and as I said, will be on open access until the end of August. So I think with that, I sometimes present the list of co-authors, but because there's a pretty long list of co-authors here, I'll let you look up the paper to see who are Isabella's colleagues. So with that, I will turn it over to Isabella and let her start her presentation. Isabella, go ahead.
Awesome. Let me just share my screen. Hopefully everyone can see that. Yep, cool. All right, well, thank you for the introduction, Daryl. And as you said, this was my honours project that I recently finished last year. And as you can see, there was a variety of scientists alike that have collaborated to make this happen, which I'm forever grateful for. But let's get cracking. So the title of the paper I'll be presenting today is The Population Genomic Structure of Killer Whales in Australian and New Zealand Waters. And just for interest's sake, this is of the northwestern Australian animals, this picture, which is quite a nice picture. So genetics can greatly assist in effective conservation management. This is largely due to its ability, more through genomics now, to produce data sets that are really high resolution and power to address a variety of questions that traditional ecological techniques may not necessarily be the best for. So you can investigate a number of parameters. These include things like population structure and connectivity. Two really good Australasian examples of looking at this type of parameters include the common dolphins. So common dolphins are widespread all throughout Australasia, or most of Australasia, and they have quite a high interaction with the fisheries industry. So being able to delineate population structure and the connectivity even between New Zealand and Australia obviously helps when looking at bycatch numbers and whatnot. And even as another example of a commercially important species such as snapper, which is kind of work that's being done at the moment, but being able to delineate stocks and their connectivity obviously helps with stock maintenance and helping prevent depletions. Some other great examples of parameters that can be used when looking at genetic, or more commonly now known as genomic data sets, is health. So some really good examples of that include the iconic charismatic koala. So koalas are actually in some places quite highly inbred, and being able to obviously understand their history is super important when looking at their conservation. So when considering things such as translocation, being able to use genomics data sets of high power gives you a great insight into how to best preserve the population. Another example is the Tasmanian devil. They are a species that can be quite succumbed to diseases such as cancer. So being able to understand the relationship between the species and diseases is obviously quite useful for policymakers when making the best policy decisions for the species. You can also look at things like how adaptable a species is to an environment. And this is obviously quite important in the face of climate change and looking at how species may persist. I think one of the coolest things about using genomic data sets is that for species that represent cryptic and even complex population histories, it can help provide baseline data that is highly resolved. So you can look at things like structure of species that may be slightly problematic to study. So population genetic structure is considered to be the spatial distribution of genetic variation within a species. So it can be affected by neutral factors such as mutation, gene flow, non-random mating, and even genetic drift. And it can also be affected by adaptive factors such as natural selection. So for species that are considered rare and elusive, this is obviously a great tool, especially using genomics to try to look at the patterns of structure within a species, because you don't necessarily need to be with the species a lot to get that type of information. So a really good example of a species that needs that sort of information and has those rare and elusive characteristics in a lot of the regions of the world is, of course, the iconic killer whale. So they're a cosmopolitan apex predator. They're found from the polar pack ice to the equator. They're currently listed as data deficient by the IUCN, and there's several ecotypes currently recognised with a single species. So as on the right of the screen, they're the current ecotypes that are recognised, and at least based on these ecotypes alone, they do seem to have distinct morphology, social structure, feeding ecology, even acoustics, physiology, etc. Generally, however, from a global basis, they're managed on an ad hoc scale, so basically just where the information is available. And this, obviously, in some cases of the world, like the North Pacific, is not generally the case, but in a lot of places like the Southern Hemisphere, there's actually quite little known about the killer whales in those regions. And this might be based just due to lack of dedicated research effort, but also probably due to their complex population history. So in places such as Australasia, for instance, we know the animals are there, but we just don't have any information yet that can really be used for effective conservation management of the species regionally. So within Australasia, they've been sighted all throughout the region. However, there's been 3 main aggregations found. So one in New Zealand, where they've been seen year round, and they've got quite a long documentation of killer whales in the region. They've also had famous strandings, such as the Type Ds in 1955. So there's a lot of, I guess, records for that area. However, more recently within Australia, within about the last decade, there's been the discovery of these 2 Western Australian aggregations. So we have one in southwestern Australia here and another in northwestern Australia. When we're looking at these aggregations in closer detail, we can see that the tropical Australasian killer whales, so those in northwestern Australia along the Ningaloo Reef, seem to have about 30 individuals identified. There is new individuals that come in and out of the area once every couple of years, so the numbers may increase. Seasonally, they do visit in austral winter, which is between June to August, essentially. However, they have been noted to be there between September and October every so often as well. There's 3 main social groups. The main social group actually is this group here. So there's a matriarch called Hook, and she has 3 offspring. Their diet mostly consists of marine mammals, particularly they're known for their predation on humpback whale calves. There is a stock of humpback whales that actually come through the Ningaloo Reef area, the Type D stock, and they calve in the area, and quite often they're known for taking calves. I think only just last week this group took down 2 calves in one day. There's also some recent whole genome evidence from a paper by Andrew Foote that was published in Molecular Ecology that suggested that perhaps these animals do have some sort of tropical ancestry. However, we obviously need to look at that in more detail, as it was only a single sample study as well. So I've got a video which hopefully doesn't lag too much, and it does have no sound because obviously it's a bit of a hectic situation, but it's a humpback whale predation with the main group that I was talking about, so Hook and her offspring, as well as I think a few of the socialites in there. So we do have a mother with her calf and also an escort in the situation, and obviously the main aim of the killer whales is to separate the calf from its mum. I will warn you, this does get a little bit gnarly, so just keep that in mind when watching it. We'll go for a few minutes, I believe. So that was Hook's that's just surfaced. So she is the main matriarch of the area and is the one that seems to be having the most calves that we know of. So as you can see, the orca have moved in and started to separate the calf from its mum. Quite often these attacks as well, they do happen quite close to shore. Not always necessarily outside, most of the time not, because I guess the visibility is better also closer to the coast. I'd also like to mention that it was John Todderdell, so the second author on this paper, that kindly is letting me play this footage of this pretty cool event. Okay, so this is just a nice video of a live humpback calf and its mother to help rectify any feelings you had towards, or bad feelings you had towards the killer whales in the video prior, but obviously it's just a natural event. Now, the other aggregation in Australia is the aggregation of temperate Australasian killer whales in southwestern Australia in the Bremer Sub-Basin. Now, just a bit about the Bremer Sub-Basin. It's a really, really cool area. It takes us about 3 hours to get out there by a boat. Obviously, it's a sub-basin, and it's a highly, highly productive region. There's so many cetaceans and other species that come through. At the moment, there's been over 200 animals catalogued of killer whale there, and we do suspect there is a fair few more that we are yet to catalogue and identify ourselves. Seasonally, they actually visit in a different time of year, so in austral summer, so between January and April. We do have some recent evidence, however, that they actually might be there longer than we suspect. The issue is that because it's in a sub-basin, it's a very, very hard site to actually work in, even in the best conditions. So it's something we'll hopefully find out more about in the future. Morphologically, the Bremer aggregation seem to be similar to type A killer whales in that they have similar eye patch positions, coloration, pigmentation patterns, etc. In terms of differences, they are a little bit smaller in size, so around 7 meters-ish, we've seen them up to, and they do have more of a falcate dorsal fin as well. Their diet, it's a bit different to that of the Ningaloo aggregation in that they do have more of a generalist approach. So mostly marine mammals, so baleen whales and beach whales, they're quite often known for fish, and they actually even think giant squid, which is pretty cool. Again, from the same single sample study as I talked about before, they may perhaps be historically genetically similar to other Southern Ocean animals. Now, something I'd like to point out is that between these two Australian aggregations is that they're in the locations at different times of year, and they've also had no photo ID overlap between the two aggregations. So it was already leading us to believe that perhaps these guys were representing two different populations, but of course we're not sure because we have only seen them for certain periods of time. Now moving on to a predation video as well from the Bremer Sub-Basin. This is actually a blue whale predation. As far as we know, it was actually the first successful predation of a blue whale recorded. We've actually got a manuscript in the review process with Marine Mammal Science, which will hopefully be out soon. And you'll be able to see more video footage and whatnot of the event. It was obviously quite a surprise, and we're all very, very excited and a little bit devastated, mostly excited. It was the middle of a lightning storm and it was raining a lot, but it was, I don't think it will be something I ever forget, not very quickly anyways. And as you can see, there's about 5 or 6 animals that are doing most of the work, and there was around 50 animals around the whale at the time. Since then, so this was in 2019, there's been another 2 attacks, and they'll also will be in the paper that's hopefully coming out soon. So the other temperate region where there's an aggregation is New Zealand, as I mentioned before. So they've been sighted all around the islands. And one of the things about New Zealand is even though they've had a really strong record of killer whales being present in the region, there's been a lack of dedicated research probably within the last decade or so. So some of the information is slightly outdated, but just keep that in mind. But from about the early 2000s, late 90s, there was 115 animals estimated for the area, and they've been listed as nationally critical basically ever since by the government. There's been 3 subpopulations proposed by previous photo ID work and preliminary mitochondrial and acoustic analysis, which suggested that There was a North Island population, a South Island population, and one that combined both, essentially. These proposed subpopulations were all meant to have different diets as well. So a generalist diet, a marine mammal diet, and a ray-based diet. In comparison to the historical similarity from a genetic perspective from this single sample study by Andy Foote, they perhaps may have more of a Northern Hemisphere background, but we obviously need to look more into their population history into the future to understand the aggregations a bit more. In terms of killer whales elsewhere, Papua New Guinea has had scattered sightings, so we know they're in the area, but there's lack of dedicated research effort out there. Not saying it won't be there in the future, but it is something we need to keep our eye on. So there was no samples, unfortunately, for us to use for our study from there, but the east coast of Australia is a little bit of a different story. So we actually, one of the co-authors from this paper, Dave Donnelly, he runs Killer Whales Australia, which is a citizen science group, and what they've been able to do is they've been able to identify from about New South Wales, which is about here, to Tasmania, that the range of Eastern Australian animals seem to be around those localities, and there's about 62 animals that have been identified, and that quite irregularly you do actually see the same animals pop up. The issue is that because they're opportunistic, it's very difficult to get the right person in the right place at the right time to also get samples. So a lot of the regions, I guess, available biological samples are based on strandings. In terms of diet, they've been seen eating a whole range of things. In Tasmania, actually, they're known to depredate off blue-eye trevally fishing boats. So that's a unique behaviour in terms of Australasian killer whales, but is obviously seen elsewhere, like with the type Ds in the subantarctic. What's something that's been really cool about Eastern Australia as well is that there seems to be these visitations from Antarctic ecotypes. So one of the co-authors, again, Dave Donnelly, he's just published a paper in Aquatic Mammals that's highlighted the presence of type B and type C killer whales actually coming into Eastern Australian waters, which is pretty cool. Now, in terms of management, there's effectively no regional management in place. There's marine reserves that overlap with some of their distribution, but it's not accounting for a lot of their distribution, so it's not heavily that effective. In terms of even the management strategies in place, basically it's just how far you can stay away from them. So in Australia, for instance, because they're a dolphin, it's 50 metres, so there's essentially no policies in place. But it does really highlight the need for baseline population data. So what we wanted to do is we wanted to look at, first of all, how many populations of killer whales there were. And because they're so rare and elusive, we wanted to use a genomic approach. So our aim of this paper was to investigate the population genomic structure of killer whales in Australian and New Zealand waters using genome-wide data. So we wanted to assess first the genetic diversity differentiation from a maternal genealogical perspective. So this is using mitochondrial control regions, and we know the mitochondrial part of the DNA is maternally inherited. And then we wanted to use a genomic data set to look at genomic diversity, differentiation, and connectivity, and number of breeders, which is also called effective population size. So we ended up with 74 samples from around a 25-year period. We had both free-ranging samples, which are from Ningaloo Reef and Bremer Sub-Basin. This is all due to the work of John Totterdale and some of my other collaborators, where they've been going out year after year for the last almost decade now. Obviously, very grateful for that. And then the rest of these samples were stranded animals. So these were from North Island, South Island, so New Zealand, Queensland, Tasmania, and Midwestern Australia. So just depending on whether we collected muscle or skin, and the quality of it depended on what type of tissue we used. But predominantly, I believe it was skin in the end. I'd just like to point out as well is that although we had a total of 74 samples that we actually used for the genetic and genomic analysis, We did actually have samples from South Australia, which is actually where I'm based, and also Victoria, but just due to the quality of them, we were unfortunately unable to use them. So going into my laboratory methods now, I'm going to go through this quite quickly and quite briefly, so please feel free to ask questions at the end or email me at a later date or read the paper if you want to find out more. But essentially, we extracted the DNA using a standard sorting out protocol, which we then quality checked using a variety of methods. If it passed the quality checking, we then prepared the genomic data by using a double digest restricted site associated DNA approach. This library was then sequenced on an Illumina HiSeq machine. Now, with these same DNA extractions, we also sequenced the mitochondrial control regions using an applied biosystems sequencer machine. Now, moving into the bioinformatics, again, I'm just going to go through this super briefly. Please feel free to ask questions at the end or email me at a later date, but just so you have an idea of the kind of process we have to go through. For the mitochondrial control data set, it's a lot simpler, if I'm honest, but we have our raw sequences from the sequencing machine, essentially. We align, we trim them, we review any ambiguities, and with a bit of magic, we end up with our final mitochondrial data set. In terms of the genomic data set, we had our raw sequences, which we then mapped to the reference killer whale genome. And we then called our markers from this genome, and this led us to having our raw SNP catalogue. So SNPs are the genomic marker we are using, which we then filtered to reduce possible errors. So these might be errors from the genotype calling stage, for instance. We then removed any outlier loci. Outlier loci in this case are the adaptive loci. So this led us to having both our neutral and adaptive data set, and we actually did not use the adaptive SNP data set at all. I'm hoping to look more into adaptive loci and things in my PhD, but for the rest of the analysis, we only use the neutral SNP data set. Now, just as a reminder, a SNP is a single nucleotide polymorph, and essentially it's just a single substitution within a sequence. So as we can see here, I've done killer whales because I like killer whales, but we've got 3 distinct individuals and they've got distinct changes or differences in their DNA sequence. So we can see that this light blue killer whale has a T and an A that's distinct to this individual. We can see the coral animal has a C and a G that's distinct to this individual. And we can see that the A and a T is distinct to this individual. So basically, at the premise of what we're doing with this genomic data is we're comparing similarities and differences of sequences between individuals to look for patterns. And in this case, the kind of patterns we're looking for is to do with structure. So heading into our results, we sequenced 72 samples in total for the mitochondrial control region sequences. And we displayed this using a minimum spanning haplotype network. So to understand this haplotype network, you just need to know that every circle represents a haplotype, and a haplotype in this case is essentially just a maternal lineage. Every colour is coordinated based on the locality, as seen here. Every line represents a mutation, and the size of the circle is dependent on how many samples actually shared that haplotype. So what we can see is we do have 5 haplotypes, so we do have these 5 maternal lineages, and because we know killer whales are characteristic for their matrilineal societies, they are matrilineal lineages in this case. And from what we can see is that most localities, or all localities, only have 1 haplotype, but there is 1 haplotype that's shared amongst several localities. So Southwestern Australia, New Zealand, both North and South Island and Tasmania share the same haplotype. What else we can see from this data is that there's little diversity within the mitochondrial sequences. Now, if you're comparing to other cetaceans, yes, it is low. However, for killer whales, it's very consistent with what's been found previously. It's important to note that it's been proposed recently, actually, that killer whales have actually undergone a bottleneck in some regions. And we know for a fact that some of the Australasian animals were included in that analysis. So this is a possible reason for this. It might also just be due to their matrilineal societies that have basically just reinforced this low diversity. However, in terms of population structural analysis, it's very uninformative, so I won't be going any more into that with this data. Now, looking at population structure with the genomic data, so just essentially how many populations are there, we ended up with almost 18,000 SNP markers, which is actually quite a lot. And what we first wanted to do was look at the overall variance of our data. So we did this using a PCA, which is a principal components analysis. And on the X and Y axis, it describes how much variation is explained by that axis. All the dots represent an individual and the colour is based on locality. And what we can see is that there's these three clusters. So we've got Northwestern Australia, South Island and North Island as another grouping. And then we've got Southwestern Australia, Queensland, Tasmania and Midwestern Australia. So due to these clusterings, obviously we wanted to work out more. Do these clusters actually mean anything? So the next thing we did was we looked at FST. FST is a fixation indices, and in this case, it's just telling us how different is this locality from this other locality. And the higher the FST, the higher the difference, essentially. So we can see from Northwestern Australia and Southwestern Australia and New Zealand, it seems to be where the highest differences occur, again supporting this idea of these three clusters, perhaps they were populations. And then we used admixture. So admixture actually tells you the most likely population or number of populations within your data set. And what it does is it basically just models ancestral proportions and the likelihood of an individual actually belonging to a population. So to understand this graphic, basically every single column here is actually an individual, and it's the percent of them belonging to the population on the y-axis. So what we can see is it does highlight this idea of these three populations, which are also highly supported. So we've got Northwestern Australia as one population, Southwestern Australia as another population, and New Zealand as the third population. So we can conclude that indeed we do have a minimum of three populations. However, something I'd like to point out is these strandings. So we did have strandings from the east coast of Australia and also Midwestern Australia, and they have been assigned to the Southwestern Australian population in this graphic. We did do additional assignment tests, and we actually saw that they had low to moderate assignment to this Southwestern Australian population. So what that really suggests is that we don't have the full picture for those regions, and that perhaps it is really just the minimum of three populations that we've found, and with more research we might come out with a few more. Now, when looking at genomic diversity of these populations, so essentially how healthy are they from a genomic perspective, we can see that the observed heterozygosity for the three populations is quite similar to other Australasian cetaceans. So that includes blue whales, common dolphins, and bottlenose dolphins. In terms of inbreeding, so the FIS is just an inbreeding coefficient, and essentially just the higher the FIS, the higher the inbreeding. And we can see there's essentially no inbreeding within the population, which is great. Although this is not really that unusual, there's only been a few cases of inbreeding recorded throughout the world in killer whales, including recently in Scotland and also a population in the North Pacific. Now, when looking at connectivity or the contemporary migration between these populations, what we can actually see is that these three populations have very, very minimal gene flow. And this is not unusual. It could just be they've been quite isolated, and it might just be ancestral remnants essentially that are coming up, or it could be that we haven't sampled all populations in the area. But again, it still does support this idea of these three populations. Now, when looking at the number of breeders or the effective population size of a population, we can see that they are generally quite low. and the confidence intervals mostly support this. I would just like to make a note that the upper interval for Northwestern Australia does seem reasonably high, but I think this is just due to the samples we've got from the population. So although we've basically sampled a lot of the Ningaloo animals or Northwestern Australian animals, I think perhaps there has been, we don't have the full picture for the area essentially yet, and we're obviously limited to what animals we can see and when we know they're going to be there. So perhaps in the future this might need to be rejigged based on any new samples that are coming into the picture. Now, in terms of what to do next for Australasian killer whale research, is that we do need to investigate the presence of additional populations, like I mentioned, even in places like Papua New Guinea, east coast of Australia, New Zealand even. We did see that the New Zealand samples in the PCA did separate a little bit from north and south, and perhaps that's just due to the number of samples we had available to us, and that worked for our analysis. I think it's also super important to look at the ancestral relationships at a global scale of these populations to kind of work out where they fit into a bigger picture, which is actually something that I'll be working on during my PhD, which is pretty exciting. And obviously research on abundance and ecotype characterization for conservation is super important. So looking at things such as morphometrics, social structure and feeding ecology will all help provide a comprehensive basis for effective regional management. Something I'd like to note as well is that I do have, there's co-authors actually in this paper, and I've got other colleagues that are actually working on some of this already, which is great. So in the next few years, we should know more about these populations and how to best conserve them. So just as a summary, we had 3 populations, or a minimum of 3 populations, so Northwestern Australia, Southwestern Australia and New Zealand. They have low connectivity. Genetically, they're reasonably healthy and they have low numbers of breeders. So what's most important is that this information is actually now available for management to start making policies around, which is really exciting, and it does put them in the picture for killer whales elsewhere in the world. I'd just like to take a moment to thank everyone that's been involved in this project, whether you're my supervisor or a co-author, and whether you helped me with analysis or obtaining permits or providing access to samples, helping retrieve samples, any of that, I am forever grateful. This project was something that really started long before I'd even started my undergraduate degree, so I'm very lucky, and I know I'm lucky to be being the one to work on this and put it together, so for that I am grateful. But besides that, I am about done. You're welcome to contact me at my email address on the top hand right of the screen, but besides that, thanks for listening.
Well, thank you, Isabella, for that interesting presentation and some rather impressive video footage of predation events. So I forgot to mention at the outset that questions should be directed to the Q&A rather than the chat, although I believe the chat will be monitored for some questions. So I think with that, I'll turn it over to Teresa, I guess, if you have questions that have been lined up for Isabella.
So at this point, we might want to give folks a couple minutes. We have one question that we can start with, and then maybe Daryl, if you have a couple, you can do those to give folks a couple of minutes. But right now we have a question of Bonnie is asking, have there been any documented instances where humpbacks try to break up a killer whale attack on other species? This has been documented on the Pacific coast in the US. Do you know of anything like that in your study area?
Not to my knowledge, no, although I will be honest, I have, compared to some of my collaborators, they've been out there a lot longer than me, but so as far as I know, no, not in that instance, not like that anyways.
And then we had someone that maybe was hoping you would clarify, you did a great job going through it, but again, the different subgroups and what they feed on and how that differs in the geographic areas.
In Australia, in Australasia?
Yeah, in your study area.
Okay, I'll just go back to maybe this one. Okay, so we've got the northwestern Australian population, and they feed predominantly on marine mammals, so they're known for their humpback predation. Sometimes we've been seen taking smaller dolphins as well, and quite often, you know, they've been in the area because everything's disappeared. There's like nothing, there's no turtles, no sharks, no dolphins, nothing. In terms of southwestern Australia, they're more of a generalist population from as far as we know. They've been seen eating baleen whales, so humpbacks, just recently actually. Blue whales, beach whales is quite common for them down there as well. A lot of fish species and a giant squid, they think. In terms of New Zealand, so that population, there's the proposed subpopulations that we've kind of clarified as one population for the moment anyways, until we get more samples. They've been seen eating a variety of things, so from rays to marine mammals to, I think they've even had cases of penguins, and more of a like generalist diet that may also include fish. I hope that clarifies any of those questions.
Yeah, I think that was great. Is most of that data coming from just observations or from stomach contents, or how is that information gathered about diet?
So a lot, two ways, and most of it at the moment has been through observations, but we do have actually a killer whale biologist here. He's doing his PhD on feeding ecology of southern hemisphere killer whales, and he'll be able to provide more information when some of his papers start coming out. But it seems to be the main things that we've been seeing, well, people have been seeing them eat on.
Great. Okay, we have a couple more questions. So Stephen has said, we usually worry about small populations having a higher chance of extinction than larger populations, and these are some really small populations. Do you think that in the long run, killer whale populations somehow manage to persist despite being small, or could there be natural high rates of population turnover or local extinction?
I think, I don't know, it's a good question. It's also a very big question. I don't know if I could give you an answer on that, but I think because of their biology, like they are the ocean's apex predator, there's not many cases of attacks on them. There's been a few, like at least in Australia, they suspect that one of the calves was taken by a tiger shark. They don't have many natural predators, and they are obviously quite adaptable to certain or a wide range of environments. So I think they do have a good chance of surviving, but obviously they're still going to be influenced by any human impacts, etc., the same as any other species. That's probably the best answer I could give on that.
That's great. We had someone asking, I don't know, this is Patricia, I don't know if I missed the data, but how many SNPs did you generate?
I generated almost 17,500. So I think it was 17,491 SNPs, which was from 51 samples in the end for the genomic data.
We have Andrew is wondering, do you think that some of these populations might represent declines rather than completely discrete populations? Just seeing that as a gradation in the pattern in the PCA and admixture plots.
Yeah, so I assume Andy will be talking about like with the possibility of, I guess, even isolation by distance. I think in the PCA it's quite clear that perhaps that if with more samples we might be starting to see that sort of pattern. So yes, they could represent clients, but I think obviously we're just limited to what samples we actually have on hand at the moment. And that's why I was a bit devastated when the Victorian and South Australian samples didn't work out as planned, because I think they may have filled in some of the gaps that are in the data set that we have now.
Okay, Alex is wondering, would you expect that within the southwestern Australian population, there could be more than one population if there was more data available? There seems to be a lot more individuals spread out along different coastlines.
So the Southwestern Australian aggregation, so just to put it in perspective, with the 200 animals catalogued, I think we've got about 60 biopsies that are from different groups. So I think at the moment, I would say they're one population. In saying that, there's been a small number of individuals from Midwestern Australia that have been showing up, and we've actually, one of the collaborators, Beck Wellard, has just had the first ID match of the same animal actually coming back into the area. So perhaps that's part of a different population, but I think, personally, I think that might not change, just based on the range of samples we've got from differing families and groups. But maybe, I don't know, it's hard to say.
Great. We had someone ask about the recording. The recording will be available after this talk, a couple days afterwards. It'll be available on the Society for Marine Mammalogy's YouTube page. And it'll also be sent out as a link to everyone who registered for the talk, for those who are asking. And Isabella, someone has asked if you can share your email again, if you want to maybe navigate back to that page.
I definitely can.
Daryl, was there anything that you wanted to ask?
I guess I would just sort of ask you, you briefly mentioned your PhD work and some follow-up on this. Can you just elaborate what directions you're taking this work, if you are, with your PhD?
Yeah, so I've literally only just started, so I'm about 5 months in. And my first chapter actually is going to be looking at population history, more specifically looking at demographic and ancestry of these Australasian populations. So we're trying to look at more at where their sort of, I guess, genomic footprints come from. Are they more related to tropical animals, or are they more related to Antarctic animals or whatnot? So that will be my first chapter, and the other 2 chapters will be based around adaptive capacity, particularly to climate change. So we should find out more about the population from an adaptive perspective as well, which will be really exciting. Well, I'm really excited anyways.
Great. Are there any other questions? Do I see one that's popped up here?
Yeah, one just popped up from Gerald, Jerry Koyman, asking, could these scattered population be remnants of past years when there were many more baleen whales?
Possibly. I don't know if everyone's familiar with the Killers of Eden. So it was the Old Tom in New South Wales, and basically his pods used to help whalers get the humpbacks that they were hunting. And after basically the humpbacks disappeared, the killer whales disappeared, and even when we saw the type D stock on the west coast start recovering, the killer whales started to come back. So possibly they're related to diet. We know nothing else really stops them. So it is likely that they are reflective of the areas. And for instance, Bremer is a really, really productive region. There's so many animals that come to the region. There's so many sharks, cetaceans, fish, even got things like giant squid. So it is likely reflective of where their prey is, I would say.
We like to ask if we have students watching, or as a undergraduate who did a lot of research, clearly, and then now as a new PhD student. It's a big question, but do you have advice or things that worked really well for you in terms of getting involved in research, especially as an undergraduate, and then continuing that into a graduate research experience?
Yeah, I think the main thing is just to try to put yourself out there as much as you can. You don't know what opportunities will come your way otherwise. I think I did a bit of that, and it's kind of worked in my favour. It's got me out on boats, and it's got me out seeing these killer whales and doing other research exhibitions. So it just, I think, as scary as it is, I'm a very reserved person. You just need to ask, as long as you're talking to people and as long as you're trying to make contact with other people and offering your time to volunteer to learn new skills, and you're going to start becoming, I guess, more desirable and having a skill set that other people might find valuable in the future.
Awesome. We do have another question from Bonnie. Do any of these populations prey on rays? I think the New Zealand orcas preying on big rays.
Yeah, so the New Zealand animals are known for their ray predations. I don't know if anyone's seen any videos, but they're pretty cool. I think they've been a few times in documentaries and things, but in terms of, I'm not 100% sure, but I have a feeling maybe on the East Coast there's been a few ray incidents. But yeah, that's as far as I know. In terms of Western Australia, I don't think so.
Okay, we'll wait just a couple minutes here to see if we get any more questions, but I think this is great, Isabella, that you know, we have had the opportunity to have you very early in your career, starting out as an undergraduate, being able to present your work to the Society for Marine Mammalogy. I think it's very impressive to have this kind of work being done by people at your level and moving forward. So any further questions coming up here? I don't see. We are approaching 10 o'clock on the East Coast of the US where I am. So I know it's morning for you there. So you're just getting started. I'm finishing up. I guess if we don't have any other questions, I'd just like to say thank you very much for a great presentation. And I do know that you have another paper currently underway in review in Marine Mammal Science. We hope you continue to be submitting papers as you progress with your PhD work. With that, I'll thank you, and thanks to all of you who attended tonight's presentation. And we have 4 more presentations lined up, I believe, for the next 4 months. So stay tuned and pay attention for what the next one will be. I don't have it in front of me, but there will be an announcement coming out at some time in the near future. Teresa, do you have anything else you would like to add?
That's great. Just a reminder, we had a couple. We had one that just came in. Stacy Fairfax was hoping to learn a little bit more if you or your advisor or your university are interested in doing more genomic research, especially on pinnipeds. So I don't know if that's something that maybe she should direct at you directly. But if you can talk a little about genomic research on pinnipeds and if there's interest in that in your university.
The marine mammal side in my university, so we, in terms of marine mammals, I believe we are, in the genomics world in Australia, we are the bigger research lab for that, but it's mostly cetaceans. I haven't seen or heard of the lab actually doing any pinnahead work, but doesn't mean they wouldn't either. There's a sea lion colony, Australian sea lion colony down there, and obviously Australian sea lions are quite endangered, and I know there's people that are other stakeholders. So one of the government departments, there's a researcher that works on them there. So I'd suggest, I'm happy if you email me if you want to know more, and I can put you on to the right people to start talking about genomics and pinnipeds, which would be pretty cool.
Great, thank you. That was awesome. Very interesting.
Excellent. Well, again, thank you very much, Isabella. You're getting all sorts of praise popping up here. So clearly lots of people appreciated and enjoyed your presentation.
Thank you for having me.
Thank you much, and you're absolutely welcome. On that, I think we'll go ahead and sign off for tonight. Good night, everyone, or good morning.